So now we're going to be studying about meiosis, and this type of cellular division is called as well reduction division. Why? Why this is called reductional? Is because remember we are going to start with a diploid cell, so that means 2n number of chromosomes, and we are going to end with 4 cells that have just half of this genetic material. So instead of being 2n, these cells will be just n. So, but before we learn about meiosis, what we need to know is what is an homologous chromosome. So one chromosome comes from the father, one chromosome comes from the mother, but if you remember during interphase, we have the stage S, and during the S phase or S stage, we have the duplication of the DNA. And is when this happens. So we make in this during this time, during S phase, the sister chromatid. So you can observe here the homologous chromosomes. And the homologous chromosomes, they have the same shape, same size, same organization of the genes, but the thing that varies and changes here may be the version of the gene. For example, in this one, it can codify for eyes color brown, but this one could codify for eyes color blue. So that is the difference between homologous chromosomes. They are not necessarily exactly the same, but they are kind of like very, very similar. So let's start with prophase. So remember, in this case, we're going to have PMAT as well, but instead of having just PMAT, we are going to call PMAT1 and PMAT2 because we have two cells divisions for meiosis. So it's very important to remember that you have to put this number 1 here, so that identifies the stage that are you at. In this case, prophase 1. So prophase 1 is the largest in of all the phases during meiosis, 1 or 2, because we are going to have 5 stages happening here. So we have leptoten, zygoten, pachyten, diploten, and diakinesis. And after that, we go to the next stage. So let's start with leptoten. And we're going to see a picture, and I will be explaining about leptoten in this picture. So prophase 1, leptoten. So here you can observe the chromosomes. They started to condense, and at the end of leptoten, the chromosomes are going to be condensed. So you can observe homologous chromosomes condense here. And we have here two different colors, blue and red. So that will help us to recognize the difference or see the difference between chromosomes. So then we go to the next stage. The next stage is the zygoten. Zygoten. In this case, the zygoten phase, called as well as zygonema, and it comes from the Greek that says paired threads. So in this case, the homologous chromosomes, they become close, so as you see right here, and they associate to this union, this link that is called the synaptonemal complex. So after that, this will form bivalent chromosomes, or called as well tetrads. It's between homologous chromosomes. So the next stage is pachyten. Pachyten, also called as well pachinema, and it comes from the Greek that means thick threads. So here you see now we don't have just the synapses, but we have the synaptonemal complex complete. And at this point, we have the crossing over. What that means, the sister chromatids from the different chromosomes have shared the chromatin between them. So they have exchanged some genetic material. And is this why when we have a brother or sister, we don't look exactly the same. Oh, just we have here, of course, an exception to the rule, because we have the twins, identical twins, sister or brothers, that they will have the same number of, the same type of chromosomes and genes. So it's because they are twins. But in general, this is the step where the um, DNA material is mixed and exchanged, and that gives us a lot of differences between our brothers and sisters. And of course, we look alike, but we are not like the same, like twin brothers or twin sisters. So the part where the chromosome exchange the material, this part right here where you see red and blue, or blue and red in this case, right here, this part is called chiasmata. So remember, when we have the complex synaptonemal complex form, we get a bivalent or a tetrad chromosome. That means that this chromosome is going to have 4 sister chromatids. 1, 2, 3, and 4 sister chromatids. The next step is diploten. And in diploten, as you can see, the synaptonemal complex is not all the way down, break or dissociated, but it starts happening. And one thing that you need to notice here is that the chromosomes that are in this part, there is still the chiasmata. So the chromosomes still linked together or glued together by the chiasmata. And that means by the genetic material that they had exchanged. So diakinesis is called moving through. In this case, the homologous chromosomes, they continue to separate, and the chiasmata moves to the ends of the chromosomes, and prophase 1 ends here. And the chromosomes now recondense, and the transcription stops, and the transition of metaphase occurs. But something very important to remember here and to know here is that the women, when they are babies, their eggs are going to be in diploten stage. So they're not going to go further than this stage. So eggs in women will be in diploten until puberty. So that means when they are become teenagers. Before that, during their childhood, or when they're babies, the eggs will stay in diploten stage. So after prophase 1, we're going to have a stage between prophase and metaphase 1. This stage is called prometaphase 1. So in this stage, the spindle apparatus is going to be formed, and the chromosomes will be attached to the fibers by the synetochores. So that happens in prometaphase 1. So metaphase 1, you see now the difference between mitosis and meiosis. In meiosis, we saw just one sister chromatid in each, and in this case, we see 4 because of the chiasmata. They link together the 4 sister chromatids, and that forms the tetrads. So in metaphase, what happens is the chromosomes are in the middle of the cell, in the center of the cell. The homologous chromosomes are paired, so they will divide 1 for each cell, where they are linked to the spindle by the synetochore. These cells, the one that we start the cell division, are diploid cells that are going to make haploid cells, so we can have just 23 chromosomes in each gamete. Remember, the gametes are sperms and eggs. So after metaphase, we have anaphase. In anaphase 1, we can see here the sister chromatids, they remain attached here, but they go to the central, towards to the centrals, to the poles of the cells. And they are going to go to the next stage. So here we have the separation of homologous chromosomes. And then next, meiosis 1. Telophase and cytokinesis 1. So, in telophase, the chromosomes become diffuse and the nuclear membrane reforms. The spindle disappears, and in the cytokinesis, the cytoplasm is divided by a cleavage furrow. The result is two haploid cells. Each cell contains one duplicated copy of each homologous chromosome pair. The cellular cytoplasmatic division is to form two new cells, and this will be followed by meiosis 2. In the case of meiosis 2, we are going to have again the same steps. PMAT, prophase, metaphase, anaphase, telophase, and then the process of cytokinesis. So in prophase 2, the chromosomes begin to condense, the nuclear membrane breaks down and the spindle forms. Then, in metaphase 2, that we have here, the spindle fibers are attached to chromosomes and the chromosomes align in the center of the cell. The next stage, anaphase 2, the chromosomes separate and they move towards to the opposite poles of the cell. So you can see the chromosomes are going to the pole. And the chromosomes in telophase 2, the chromosomes reach the pole ends and the nuclear membrane reforms. And then we have the cytokinesis. The cleavage furrow divides the cell into 4 haploid cells. So after cytokinesis 2, we are going to obtain 4 cells, and these cells are haploid cells. So instead of having 46 chromosomes, they are going to have just 23 chromosomes each. Remember that in the case of male, each cell is going to produce 4 sperms, but in the case of women, each cell is going to produce 4 cells, but just 1 egg and 3 polar corpuscles. So just the egg is the one that is going to be fertile, and the one that can make with the fusion with the sperm can make a new baby.